What's up, IB Bio students? This is part 3 of 3.4. So your warm-up for this time is going to be to construct a Punnett square to explain this occurrence and this happening. This is a picture of Gregor Mendel. He's the founder of genetics, and he specifically discovered Mendelian genetics, which is that recessive dominant allele inheritance pattern thing that we talked about before. So we've got here one parent that is a male, one parent that is a female, and these are pea plants. And so the yellow represents the peas that are yellow, right? And so both parents are yellow, and they ended up having offspring, and their offspring turned out to be 75% yellow and 25% were green. So the question is, how did this happen, and could you explain it using a Punnett square? Pause the video and try to figure this out, and then I will give an explanation. Okay, so the example that we were looking at had two parents, both were yellow, but they produced 25% of their offspring that were green. So let's take a look at what this would look like with the Punnett square. Remember, we were using big Y to represent green. We were using little y to represent yellow. So we're going to draw the alleles of what this would be on a Punnett square for each parent. So you've got one parent. If you thought that it was one parent that's heterozygous and another parent that's heterozygous, you would be correct because it would look like this. And remember, you always draw the capital letter first when you're doing the Punnett squares. Okay, so that's what it would end up looking like. And the phenotypic ratio would be a 3-to-1 ratio, because as we can see here, this is a 3-to-1 ratio. 75% yellow, 25% green, right? The genotypic ratio, remember, the genotypic ratio would be 1 to 2 to 1. Now let's look at this one. We've got two parents. They haven't filled in the colors on them yet, but we do see a description of their genotype. So that's homozygous recessive for the mother, homozygous recessive for the father. Homozygous recessive should mean what are the alleles of both of the parents' genotypes. It should be both of them have little y, little y, little y, little y, which is green, right? And so if you would pause your video right now and try to actually do the Punnett square for that as well, and then I'm going to say the answer. Okay, so the answer would be that you've got little y, little y, little y, little y, and you end up all with offspring 100% are going to be green because they all have homozygous what genotypes? They all have homozygous recessive genotypes. And so the phenotypic ratio is 100% green. Go ahead and practice this one as well. Here we have a homozygous recessive mother being crossed with a heterozygous father. Okay, so you're going to have to think of what are their genotypes. You're going to have to put those into the Punnett square. And by the way, these squares on top where the parents are, each of those squares represents their gametes. So over here would be for the father, one sperm, and then another possible sperm that he could make. Then over here, this would be one possible egg with the allele in the egg, and another possible egg with the allele in the egg of the mother. Okay, so pause your video. And as you can see, this is what it should look like if you were to complete the Punnett square. We've got heterozygous for the father, homozygous recessive for the mother, and it ends up with a 1-to-1 ratio in the offspring. So we've got half of the offspring that are yellow, half of the offspring that are green. And then the genotypic ratio would also be 1-to-1 because we've got these heterozygous offspring right here, the two, and then the two homozygous recessive offspring over here, and it simplifies to a 1-to-1 ratio. Okay, again, you can pause your video and try to complete this one, and this is what it should have looked like. You can see that the genotypic ratio, is it different than the phenotypic ratio? The phenotypic ratio, you end up with 100% yellow. But the genotypic ratio is different. We've got homozygous what over here with half of the offspring? Homozygous dominant. Over here we have what with the other half of the offspring? We've got heterozygous. Okay, so if you remember the essential idea of 3.4, it's that the inheritance of genes follows patterns. So far we've talked about Mendelian inheritance patterns with simple dominant and recessive alleles. It gets more complicated than that. So this is codominance, an example of codominance, but it also has a recessive inheritance pattern as well. It's with blood types. So of course you all know about blood types, and a lot of you probably know your own blood type. But do you know how to look at your parents' blood types and see what the chances are that you are the blood type that you are? It can be kind of a fun activity to do. So go ahead and ask your parents. You can even ask your grandparents what their genotypes are and see if it matches up with what your parents are. If it doesn't, then you've got some problems on your hands possibly, or somebody was adopted somewhere along the way. But you can figure out the chances, the probability of you actually having your blood type if you know your parents' blood types. So first, to understand how all of this works and how to use a Punnett square to predict the probability of you having your blood type, you definitely need to know your parents' blood types, but you also have to know about the different blood types, right? And what they all are. So the different blood types are shown here in the blue on this chart right here. You can have either type A, type B, type AB, or type O blood type, right? And with each of these blood types, you also have things called immunoglobulins that are on the outside of the RBCs or red blood cells, okay? And so the immunoglobulins would be something like the antigens. And so the antigens are actually proteins that are out on the surface of your RBCs. So blood type A has A antigens on the outside of the blood cells. Blood type B have these antigen B proteins out on the outside of the blood cells. AB blood type has antigens A and B on the outside of the blood cells. And then something interesting about O is they actually don't have any of these antigens on the outside of their blood cells. Generally, what antigens are used for is just a marker, sort of a tag to say what type of cell you are and to identify the type of cell as either a cell that should be in the body that is a part of your body and your body's specifically related a lot to the immune response, and that, or to say, this isn't a part of my body. For example, if you put a blood type B person's blood into a blood type A person's body, the A person's body is going to react negatively to that because your body would be recognizing those antigens on the outside of the B blood cells as foreign cells that don't belong in the body. Okay, and your body does the same thing with pathogens, including viruses and including some bacteria as well that have antigens of their own on their own cells or on the capsid of the virus possibly. And so your body will recognize those antigens as either being foreign antigens or antigens that are a part of your own body. Okay, and so part of, and we can talk more about this later, but antibodies that are produced by blood type A people, antibodies, not antigens, that are produced are antibodies for B, the B antigen. And so essentially, these antibodies tell a person with blood type A, these anti-B antibodies would tell the person with blood type A it would tell their immune system to sort of attack these antigen B blood cells that may be introduced into the blood type A person's body. But anyway, don't worry so much about the antibodies. I wouldn't worry so much about that. Or even the antigens, you don't have to worry that much about. What we're worried about right now is this inheritance pattern called codominant inheritance with blood types. And so the genotypes for these different blood types, this is what the genotypes look like. They're very different than what we've looked at with lowercase and uppercase letters. Here we've got, yes, we have a lowercase letter, and this one is the O type blood allele. Okay, and it is recessive, which is why it's a lowercase letter. But then here we have uppercase with a superscript A, uppercase with a superscript B, and this is the A blood type allele, and then the B blood type allele. And if you look at the genotypes of the different blood types, you can see for blood type O, what is the genotype? It's going to be little I, or lowercase I. And then for A and B blood type as well, they can actually have two different types of genotypes. So we've got uppercase I superscript A, uppercase I superscript A. So it could be two uppercase I's and two with the superscript A. Or it could be one uppercase I with a superscript A and a lowercase I. Do you see how that sort of makes sense that it's still going to be blood type A? It's because that superscript A is actually overpowering and the uppercase I is represented as the dominant sort of allele that's overpowering this lowercase I recessive allele. And so blood type O just has two of the recessives, and that's how you get O blood type. And then over here with B, it's the same situation as with A. With AB, this is where the true codominance comes into play that I was talking about before. Here you've got blood type AB, and it's because both of these superscripts, superscript A and superscript B on these uppercase I's, both of them are actually being expressed in your blood type. And so this is called codominance, when both alleles in the genotype are expressed. Okay, so let's take a look at if you have blood type B and you want to know what the percent likelihood was of you getting blood type B if your parents have what they have. So what we're going to do is first identify all of the alleles and the blood types that go with them. The only alleles that can produce a blood type O are lowercase I, lowercase I. The alleles for blood type A, let's do that. The only ones that can make blood type A are uppercase I with the superscript A, another uppercase I with the superscript A, or you can also have uppercase I, superscript A, and what? Lowercase I. Why can you have that second one? Because if you have the lowercase I, remember that's a recessive allele. So this one is more dominant, the uppercase I, so it's going to cover that, dominate over the lowercase, and you end up with blood type A. Same thing for blood type B, same situation as blood type A. So you've got uppercase with the superscript B, or uppercase I with the superscript and the lowercase I, and then you end up with blood type B. And then the last blood type, of course, is AB. And there's only one possible genotype for AB, and that is going to be I with the superscript A, I with superscript B. Remember, that is what type of inheritance? That's where you see the actual codominant inheritance right there.